Good afternoon, everyone. I am Dr. Swati, Assistant Professor in the Department of Food Technology, Haldia Institute of Technology, and today I am going to present my lecture on Escherichia coli detection in food sample. So before we start with the main topic, we need to know about the group in which Escherichia belong, and that is the most significant group in food sample, which is called the coliform group. Now, what are these coliform groups? The coliform group are the group of bacteria which are principally present in the digestive tracts of warm-blooded animals and human beings. So, since they are present in the intestine and gut, mostly they are excreted through excreta and feces of this animal. So when they are excreted through the feces, they contaminate the nearby water body, causing water pollution, and also they release their spores into the water system. Beside the digestive tract, they are also present in various plant materials, in soils, and abundantly distributed in the environment. Now let us discuss about the morphological characteristic of these coliform groups. Principally, these groups are aerobic in nature. They are gram negative, that means they stains negative in the gram staining process, and they are non-spore formers, rod-shaped structure, which is designated as bacilli. Now, some of them are motile, that means they have some locomotory structure to move from here and there, principally flagellum, or some are non-motile as well. Now, when you try to grow them, their optimum growth temperature is around 35 to 37 degrees Celsius, and they possess a particular intracellular enzyme which is beta-galactosidase. So due to presence of this beta-galactosidase enzyme, they can ferment lactose to produce acid and gas. Now, among this coliform group, the principal organism of significance is the Escherichia coli or E. coli, because it is used as a monitoring device or monitoring microorganism in food system. Now let us discuss in details about the significance of this coliform group as well as E. coli in food system. Now, E. coli is distinguishable from the other coliforms since it can ferment lactose at 44 degrees Celsius, and it is most of the time used as an indicator in the sanitary quality maintenance of food in dairy industry and in water. Most of the strains of E. coli are non-pathogenic. They cause very mild symptoms if their number increases in our body, but some of them can be virulent, some of them can be very, very pathogenic in nature. So when E. coli infection happens in our body, principally it is called colitis. So in the early stage or mild stage of colitis, we may have bloody diarrhea, vomiting, stomach cramps, and fever. Now, some of the associated symptoms can be pneumonia, it can be respiratory illness or urinary tract infection as well. Initially, this particular organism was not taken very seriously, but since 2021 there were severe reported cases of foodborne outbreaks in the US. So in 2021 many people got this illness, they got the outbreak by consumption of contaminated spinach, from where the E. coli virulent strain of O157H7 has been isolated. It is a highly pathogenic strain, and just 1 year after that, in 2022, again foodborne 2 or 3 foodborne outbreaks were reported in the US itself, where the same strain of E. coli has been detected in kimchi products. Now, after these incidents have taken place, every regulatory body has mandated the detection of coliforms in their testings. Okay, so the detection of coliforms involves the detection of 4 different sets of techniques. First is the detection of total coliform, which is a bit lenient test, that if we are testing if coliform is present or not. Then the detection of fecal coliform, that means the coliform which are originating from the feces, which have a tendency to become virulent. Then we have detection of E. coli, which is the principal coliform, and it is used as an indicator organism for the presence of other pathogenic microorganisms. And we have pathogenic E. coli. So pathogenic E. coli means we can have strains like E. coli O157N7. Now, for detection of the coliform group as well as E. coli, we can perform some biochemical tests as well as we can perform the microbial tests. So microbial tests are the most preferred tests because they are easy to apply, they give distinguishable result and are cost effective. So let us start with the detection of the coliforms using biochemical tests. So there are a set of 4 different biochemical tests that are used in case of coliform detection, especially those coliforms which are enteric in nature. They belongs to the group Enterobacteriaceae. Okay, so this 4 set of tests, they are cumulatively they are called IMVIC tests. So this IMVIC is an acronym for indole test, methyl red test, Vaux-Prosker test, and citrate utilization test or citrate test. So I will give you the details of how these tests are performed. So right now, indole test is a test which is done in a tryptophan broth using an indicator which is called Kovacs indole reagent. Methyl red test is done in a specialized media which is called MRVP broth or methyl red Vaux-Prosker broth, adding methyl red as the indicator. Vaux-Prosker test is done in the same MRVP broth with added alpha naphthol and KOH, and citrate utilization test is done in a special type of agar which is called Simon citrate agar, where the indicator is bromothiamyl blue. Now in this picture we will discuss in details about the IMVIC tests and how this test helps us to detect the coliforms in any water or food sample. So the first one is the indole test, where the sample that is to be tasted is taken and it is inoculated in a tryptophan broth. So this tryptophan broth will naturally contain lactose as its major constituents. So this lactose will be fermented and there is a product which is called indole that will be formed. So in the picture we can see that the sample which contains the coliform, as soon as we are giving Kovacs indole reagent on this broth, due to presence of benzaldehyde, it will form a red colored ring-like compounds which is detectable and which indicates the presence of certain coliforms in that sample. Next, we are going to discuss about the methyl red test. I told you previously that methyl red test is done in a special type of broth which is called MRVP broth or methyl red Vaux-Prosker broth. So this methyl red Vaux-Prosker broth is taken and the sample is given and it is incubated for a certain period of time. After incubation is over, the methyl red indicator is put into the sample. Now due to if there is a coliform present, it will produce acid. So due to production of this acid, if there is acid, methyl red will turn red in color, indicating the presence of certain type of coliform. Next, the third test, which is called the Box-Prosper test, it uses the same MRVP broth, only it is using a different testing component. So when there is a certain group of coliform present, there is an intermediate, that is acetyl methyl carbinol, is formed in the broth after incubation. So we will detect that acetyl methyl carbinol in this broth. So after it is incubated, the sample is taken and it is added with alpha naphthol and KOH solution. So if there is coliform present, then it will produce this acetyl methyl carbinol, which will react with alpha naphthol and KOH to give a pink colored solution. Whereas in the absent test, there is no such color change. So lastly, we can talk about the citrate test, where basically we are using a agar medium which is called Simon citrate agar. So if you check the composition of Simon citrate agar, it is containing bromothymol blue as the normal constituent indicator. So whenever we are growing a sample which contains certain type of coliform, after incubation, if there is that coliform present, it will turn from green to Prussian blue. So why this change is happening? This change is happening because coliforms in the process of fermenting lactose will produce carbon dioxide. So this carbon dioxide will be utilized in two ways. The first way is it will react with water to produce carbonic acid, as well as it will react with the citrate present in the media to give rise to sodium carbonate. Okay. So sodium carbonate is alkaline in nature, so it will change the pH of the medium. Another reaction that will happen is the carbon dioxide will initiate the breakdown of the ammonium salt that is present in the agar. So this ammonium salt, when they will be break down, they will give rise to ammonium hydroxide. So this ammonium hydroxide is again alkaline in nature. So due to this change in pH to alkaline range, the indicator that is present in the agar media will change from green to blue. So thus we can see that through color change and through biochemical test, we can easily detect the presence of coliforms in food and water sample. So these are the some of the list of coliforms, principal coliforms, which gives different response to immune test. Like the principal organism Escherichia coli, it gives positive result in indole and methyl red test, but it gives negative result in VP and citrate test. Whereas Enterobacter is just the opposite. Enterobacter and Klebsiella both gives negative test in indole and MR test and positive test in VP and citrate test. Now if you want to detect Salmonella only gives positive test for MR and citrate, and the two tests are negative. Whereas if you want to isolate Proteus only give negative test to VP and all the other tests are positive. So like this, through permutation combination, we can actually identify a particular coliform in a food or water sample. Next, we will discuss about the microbial tests, which are much more confirmatory and are used as a routine test analysis in different regulatory systems. So the most common test which is done is the MPN method, or most probable number method. Most probable number method relies on presence or absence test. That means it will give you a detectable change that whether the microorganism is present or absent. Okay, so under this MPN method, we have three sets of tests, which are presumptive test, confirmed test and completed test. So in presumptive test, we will be using McConkey broth. So in McConkey broth, we are putting the sample and we are incubating at 35 degrees Celsius for 24 to 48 hours. After the presumptive test, we will retest the sample with confirmed test using BGLB broth. BGLB is brilliant green lactose bile broth, and for 35 degrees at 35 degrees Celsius for 24 to 48 hours we will incubate the sample to see the result. And lastly, we will have completed test. Completed test can be done in a special type of agar media, which is EMB agar or eosine methylene blue agar, which gives distinctive colonies, greenish colonies of the coliform that is to be detected. We will discuss this test in the next slide in detail. Now, if we want to detect fecal coliform, which are much worse than the normal coliforms, total coliform, the first test that I have told you is mostly for total coliform detection. So, fecal coliform detection involves the use of EC medium, which is used for the test sample detection at 44 degrees Celsius. It is incubated for 24 hours to check any result. Now, after all these microbial tests are done, if we want to visually confirm that the coliforms are present, we can always perform gram staining method, which will give a distinguishable result that there is a gram negative non-spore forming bacillus bacteria present. Now, let us discuss in details about this tests. So first, let us see the first diagram where we have discussed about the presumptive test. So this presumptive test consists of a broth which is inoculated with our grievance sample. The broth is called McConkey broth. It is a special type of broth which contains lactose. Now, inverted Durham's tubes, which is used for gas production in microbes, is kept inside the test tubes, and these are incubated for 24 to 48 hours. After 24 to 48 hours, if your sample contains coliform, it will be turbid. There will be gas that is trapped in the Durham's tube, and it will eventually change color from red to yellow. So this color change is due to the fact that the pH is getting lowered, and the gas detection in Durham's tube is indicative of the coliform which is releasing carbon dioxide in their metabolic process. Now, this ends the presumptive test. That means we can say that okay, coliforms may be present. So after that, the inoculated tubes which has given the positive results are taken and the confirmed test is performed. For confirmed test, we are using a different broth which is called BGLB broth. In BGLB broth, which is green in color, we will put the sample and we will incubate it at 35 degrees Celsius for 24 to 48 hours. So again, after the incubation period, we will see that the turbidity of the sample that is containing the coliform, whereas the sample which does not contain any coliform will be clear. So this is the confirmed test. That means the coliforms are definitely present. And finally, if we want to check whether the colonies of the coliform, what type of coliform is there, what is the nature of the coliform, then we will complete this microbial test with the help of completed test, the third set of tests. Here we will use specific detection agar like EMB agar, where it is used for detection of E. coli or other coliforms as well. So we'll inoculate the agar plates with the sample and we'll keep them at 35 plus minus 2 degrees Celsius for 48 hours. So after 48 hours, if there are green shiny colonies with metallic shine and dark centers are found, that means there is E. coli present, as you can see in the figure. Now, if you want to see them visually, you can always perform gram staining, and the gram negative organisms, that is E. coli or the coliforms, will be stained pink and they will look like a rod shape under the microscope. So for testing fecal coliform, you can take the sample and you can grow it in the EC broth medium. the temperature is 45 degrees Celsius for 24 hours, and if there is turbidity, that means coliform is present. Also there will be gas detection in the Durham's tube, and if there is no turbidity or no gas detection, that means the solution is negative, there is no coliform present. Now when we want to visually detect Escherichia coli in specific tests, there are three ways by which it is done mostly. The first way is, I have already discussed, that we can plate the sample in specialized agar media, which is using methylene blue agar. So after incubation to 48 hours, we can see green metallic colonies, and we can see the colonies have dark centers. There is a drop of bile that is precipitated in the media as well, so like you can see in the figure. Another agar media which is often nowadays used is called violet red bile agar. Violet red bile agar also detects E. coli, but the nature of the colony in this agar is like pink mucoid colonies with white colored bile deposited at the end of each colony. Now whenever we are doing this microbial agar test, we are taking a very small amount of sample. Now if we want to detect the total coliform for a large amount of liquid sample, then there is a membrane filtration technique that is used. So in this technique we are using a membrane filter where the media is absorbed in the filter, and the whole sample is filtered through that membrane. So during filtration, all the coliforms will be entrapped into the filter matrix, and since the media is already absorbed in the filter, it will be incubated, and growth of colonies can be seen in the membrane filter itself, like you can see in the slide. Okay. So beside this easier and cost-effective biochemical and microbial tests, we have other high-end tests which are much more less time consuming. They are quick, they perhaps takes half an hour to 10 minutes for detection, but they are highly costly. That these tests are also used for detection of specific coliforms in different food and water samples. So these tests include polymerase chain reaction or PCR techniques, chemiluminescent in situ hybridization techniques, where we are detecting 16S RNA for 546 BP molecular weight DNA. Then we have epifluorescent microscopy for direct detection of the coliform. We have enzymatic assays, we have ELISA tests, we have immunofluorescent assay, we have immunomagnetic separation. So all these techniques are advanced techniques, they take less time and they are used for detection of coliform very quickly. Now concluding, we can infer that the coliform group has a high significance in food safety, and it is well distributed in the environment, in water, in soil, in plant materials, as well as in the gut of humans, causing intestinal diseases. So this E. coli, which is a coliform, can be used as an indicator organism for detection of other pathogens. So why we are doing this indicator organism test? Because most of the cases individual pathogen detection is very costly. So if we have an indicator organism which is detected, so we can infer that there may be some pathogens that is present. So the person who is taking care of that unit, they can detect the source from which this contamination is happening, and they can restore the hygiene of that particular system. So with a very less cost, we can easily perform this experiment. That is why indicator tests are very much preferred in regulatory systems. So as we have seen that most of the E. coli they are harmless, but there are some pathogenic strains like E. coli O15787 which actually may cause foodborne illness. So this detection is mostly done by traditional method and by biochemical test, that means microbial test and biochemical test, but advanced techniques are also there for detection with a very less time. Thank you for listening to my lecture.